Hey everyone, so we're going to get into how we connect those gametes that we've talked about in meiosis to inheritance and how those genes actually get passed along today. So take a moment, look at the learning goals so you have an idea of what to focus on for this lesson, and then press play when you're ready to continue. A couple of words that I want to go over just so we have an understanding of what these are going to mean as we're going through the lesson. So one is phenotype. That's talking about the physical appearance, our trait itself that we related to those alleles. So it doesn't have to do with what our genes are saying. It has to do with how we physically appear. So I think phenotype, physical appearance. Our genotype is related to what our genes actually code for. So what kind of alleles do we actually have? The two of them are not always exactly the same thing. Hopefully that word looks familiar. So anytime we're talking about homo, we're talking about the same. Zygous is talking about that zygote that is formed when we first get the sperm and the egg come together to form that first cell. So homozygous is referring to any kind of offspring that have two of the same alleles inside. We've got two examples of that in the picture in the corner here. In this picture, we have two blue alleles that are represented. So each chromosome, one from mom, one from dad, each of them had a blue allele. And so it's homozygous for blue. This one's homozygous for brown because both of those chromosomes have alleles for brown eyes. Hetero means different. So when we're looking at something that's heterozygous, that means that it's going to have one dominant allele and one recessive allele. So as we look here, we've got one blue eye allele and one brown eye allele there. One thing to just keep in mind, we're talking about homozygous and heterozygous. We're not talking about gametes. We're talking about the parents, or we're talking about the offspring, because none of the gametes should have two of the same trait allele in there. So we should never have a gamete that has two alleles for eye color, because those things should be separated out. Gregor Mendel was the first major scientist when it came to genetics, and a lot of what we understand now is because of his early understanding with pea plants. He chose these because they were really fast to breed, you could pollinate them easily, and there were a bunch of traits that were really easy to be able to tell the difference between, so tall versus short, or yellow versus green pea seeds. For his first experiment, he took plants that had been breeding for generations, and they always created tall plants. And so that's why we call them pure breeding plants. Now, pure breeding plants would have to be homozygous, because for them to be able to continually create the same phenotype, the same appearance, they would have to have the same alleles that they're passing on every generation. So tall plants in that group only ever had tall alleles. In another group, he took dwarf plants that kept on producing dwarf plants for generations. Those were also homozygous, because if they kept on having dwarf plants, that meant that the only allele that they'd be passing along would be the dwarf allele. Now he took one of those tall plants that originally just kept on producing tall plants and one of those dwarf plants that was also pure breeding, and he crossed them together in a parent generation. So just on the side there you see P for parent. Those are like when we're looking at our first gametes that are being introduced into a cross. So he had a tall plant, he had a dwarf plant, and something really interesting happened in that first generation. All of the plants were tall. So that dwarf plant, it seemed like that one disappeared. We refer to a dominant trait as the trait that takes over in a situation. So in this situation, tall would be the dominant trait because the tall trait took over the recessive trait. Each of those plants would have to have one tall allele and one dwarf allele, because that tall plant could only give tall alleles, that dwarf plants could only give dwarf alleles. However, even though they were heterozygous, they all looked tall. So that means that that tall allele dominates and takes over from that recessive allele. In the second experiment, he took those same plants from the first generation, so the ones that had one tall and one dwarf allele, and he crossed those together. And we actually see that recessive phenotype pop up again. So here we had 75% of the plants that were tall and 25% of the plants that were dwarf. So in that first generation, that dominant trait took over and that recessive dwarf plant was hidden. But in the second generation, that phenotype popped up again. Now, we call these monohybrid crosses. mono meaning one, because we're only looking at one trait here. We're looking at the height of the plant. We have something called the principle of dominance, which states that if you happen to have a hybrid, so someone who has two different alleles, one dominant and one recessive, then only the dominant trait will appear. This is an example of what we call complete dominance, in which the tall allele completely dominates over the recessive allele so that we don't see any recessive allele whatsoever. We only see the tall one or the dominant one. We represent this dominance with a capital letter. So I'm going to use T here since we're talking about the dominant being tall. So I use a capital T to represent the fact that that is the dominant allele. When it comes to the recessive allele, we use the same letter. So I want to make sure that I'm using a T for both the dominant and the recessive allele because they're the same trait. I'm talking about the height of that plant. But I'm going to be using different forms of that letter to represent dominant versus recessive. So dominant is your capital letter, recessive is your lowercase letter. Remember when we talked about that law of segregation earlier? We were talking about how chromatids that were the same chromosome, so let's say a number 15 and a number 15, those ones would always end up separate. So we only have one set of chromosomes in each gamete. Here, since we're talking about alleles, and alleles are on chromosome, it's the same idea, where we're going to have alleles always be separate. So if you're looking at gametes, you should never have it so that you would have a gamete, like this one, that would actually have two alleles of the same trait inside of it. That would be wrong. You always want to have it separate, like this. So we've separated those alleles out into different gametes. What this little visual shows us is that we have eggs from the mom and sperm from the dad. Now, that first pea plant that we had was homozygous dominant, so we have two capital T's here. And that second plant that was crossed was homozygous recessive, so we have two small t's here. The way that we look at this is that we put our gametes on the outside because either of these eggs could combine with either of these sperm to produce offspring. So this little box here, this square, gives us an idea of the probability of different sperm and egg combinations. And the way that we show that is we drop whatever letter happens to be coming from that parent above into all of the columns underneath. So we take that T and we drop it in each one of those boxes. And from the second parent, same thing, except we're going to add that allele into all the boxes within the row that that gamete is in. What we really get at is that this is one offspring that is created from that dominant T allele from the mom and the recessive T allele from the dad. Each one of these represents offspring and the probability of getting these offspring. Now, since the mom only has dominant alleles to offer, and since the dad only has recessive alleles to offer, all of the offspring have the same genotype, that heterozygous capital T, lowercase t. And all of them happen to be tall because that T allele takes over, so we don't see any dwarf. Something's going to be different in that second experiment because we have different alleles from each parent now. So same kind of thing that we did before, where we take the alleles and we're going to drop each of these letters into what's below it. In this one, we happen to have one that is homozygous dominant. We have two that are heterozygous, and we have one here that is homozygous recessive. So a little bit of a different ratio than we saw before. What's really different here is that our phenotype ratio is going to be completely different because all of these are going to be tall. So even though the genotype might be different, one could be homozygous dominant and the other one might be heterozygous, though all three of those are going to be tall. Also notice that the only time we actually see this recessive phenotype is when we have both recessive alleles. So anytime you see somebody who has blue eyes, you know that they're going to have two blue eye alleles because blue eyes is recessive. But if someone has brown eyes, you're not going to be sure whether or not they're heterozygous or whether or not they're going to have two dominant alleles. So take a moment, go through these key questions to see how much you understood from this lesson. And we're going to continue talking about monohybrid and the next one, dihybrid crosses, tomorrow.